In this lesson, we'll describe some of the key events in the early history of life on Earth. In previous lessons, we've noted the tremendous size difference between complex cells, as you see down here, small species of amoeba, and bacteria. Bacteria are the smallest and simplest types of cells on Earth. All animals and plants and fungi are multi-celled, but the individual cells are complex cells, like this single-celled organism here, the amoeba. So we noticed the tremendous size difference, and that means then that the origin of life involves a story where it's the smallest, simplest types of cells that first emerge, something like bacteria, and then we get somehow this transition to larger, more complicated cells. And one idea that's getting some attention is that life began underneath the oceans in these undersea volcanic vents. But first, let's step back a moment and take a look at the Drake equation up here. And you'll recall we studied sort of the N represents the number of communicating civilizations in our galaxy. And the F sub L term would be the fraction of planets where life emerges. Well, for life to emerge on a planet, it's got to be then an example of spontaneous generation. Non-living things are going to be producing the first living thing. Life on Earth is cellular, and so here we have our sort of cartoon version of a simple type of cell. DNA would be the key to life on Earth. It stores the recipes to build proteins, specifically how the DNA instructs the cell how to stick amino acids together to make any particular protein. And one idea that's getting a lot of attention today is that life emerged some 4 billion years ago underneath the oceans in these volcanic vent structures. One of the attractive things about these volcanic vents is they have lots of tiny little chambers. There is a source of energy, the heat and chemicals coming up from the Earth's interior, and these little chambers would concentrate lots of molecules where lots of complex chemistry might be going on. So you get almost a cell-sized compartment with lots of interesting chemistry happening here. So some scientists believe that the first living cells were hatched in these rocky nurseries. And that, of course, would mean a really weird thing, that our planet, our rocky planet, could produce a living thing. That makes our Earth one strange rock indeed. To connect up this organism here, then, that's the way we're going to think about the origin of life here, these first cellular organisms. Now, we give the acronym LUCA, the last universal common ancestor, to that living cell that was the ancestor of all living things today. There might have been some cells that existed before LUCA that were alive, but they just didn't leave any descendants that we see today. So while LUCA might not have been the first living thing, it's the last universal common ancestor. All living things on Earth could trace their ancestry back to this simple cell. So our story then is one where we have the origin of life, and lots of scientists are still studying those conditions and how it might have happened, where it might happen. So we've got some bacteria starting the show on Earth, and then something big happens. Somehow cells get big. Of course, today, bacteria remain small, simple types of cells, but there are other cells, we call them complex cells, that are much larger. And so that part of the story has to be told. Now, for a long time, this was part of the mystery of life on Earth. But a lot of work now has been done, and scientists are fairly confident that they know at least part of the story. And part of the story involves the mitochondria over here inside these complex cells. It turns out that mitochondria have some interesting properties that give a clue to the origin of mitochondria and the origin of big cells on Earth. So let's return to this diagram we noted in an earlier lesson where we were comparing the sort of simple bacteria type cell with a complex cell. And they have lots of similar features. The complex cell has a nucleus, meaning a membrane around the DNA. And of course, complex cells have mitochondria, and bacteria don't. And of course, there's the size difference as well. Well, it turns out that mitochondria then possessed several characteristics that illuminated the history of life on Earth. Now, in the late 1800s, a biologist, Richard Altman, was using microscopes, looking at animal cells, and he noticed these little sausagey things inside cells, and various stains and things would make them more visible. And this is a kind of a drawing that he made from what he saw in the microscope, and we're going to call these then mitochondria. Now, at the time, scientists didn't know exactly what they were and what their function was. Today, we think of them as the power plants of the cell. Mitochondria are where glucose is metabolized to get energy for the cell. The top picture here is an electron microscope picture, a cross-section through a mitochondria. It's got lots of membrane folds in it here, and of course there's all kinds of proteins doing complex chemistry here to break down glucose and package that energy into ATP molecules. And here's another electron microscope picture of mitochondria. Now, there are many mitochondria in a cell, as you can see from this kind of picture here. But let's just zero in on the mitochondria now. The first feature that intrigued biologists was well known after just the early studies of mitochondria, and that is their size. As it turns out, mitochondria are roughly the size of bacteria cells. There's a second interesting feature about mitochondria that was learned in the middle of the 20th century, and that is mitochondria have DNA. They have their own DNA. So let's add that. So number one, mitochondria are the size of bacteria, and mitochondria have their own DNA. In fact, it's a circular piece of DNA inside the mitochondria. Well, where do we see circular pieces of DNA? In bacteria cells. Right? So bacteria cells typically have a circular chromosome, very much like what you find in mitochondria. And of course, we know that bacteria can divide. They duplicate their DNA and then they divide into two daughter cells. Well, as it turns out, another interesting feature of mitochondria is that they too reproduce within the cytoplasm of the larger cell, of the complex cell. In fact, over on the left here we see a dividing bacterium, and you can kind of see the notch there. That's where the cell division will happen there. So, bacteria cells grow, and then they divide. And when they divide, they're smaller again, and then they will grow and divide. Well, in the picture over here, we kind of see some of the mitochondria are kind of long sausage shapes, and the idea is that mitochondria would be ready to divide at some point in the near future. So what we've got then is that in these large complex cells, we've got power plants, right? Glucose metabolism producing energy. Well, the power plants can reproduce, and that means the big cells will have lots of energy to be big and complicated and to have active lifestyles. Now, in a cell like an amoeba, when an amoeba is going to go through cell division, it will have to duplicate its DNA and then deliver DNA to both daughter cells, but it's also going to make sure each daughter cell has mitochondria to power the chemistry of life. So though we can't see the mitochondria very well in this picture, as the amoeba is dividing, many mitochondria will go to one daughter cell and lots of other mitochondria will go to the other daughter cell, so that each daughter cell will have the energy to power the chemistry of life. So with these three clues, one, that mitochondria are the size of bacteria, they have circular DNA like bacteria, they reproduce inside the larger cell like bacteria can reproduce, it started to shed light on the origin of mitochondria. In the 1970s, scientists started to propose that mitochondria were once free-living bacteria that somehow got themselves inside of a larger living cell. So now let's tell the story of some of the early events in the history of life. Some 4 billion years ago, somewhere on Earth, underneath the oceans, some rocky chambers released a living thing. And we'll think of this bacteria cell here as sort of like Luca, the last universal common ancestor. Well, Luca already possessed DNA, the genetic material for life on Earth. So Luca had the capacity to reproduce, and because new mutations happen every time DNA gets duplicated, we've got the possibility for evolution. So bacteria, as they reproduce, survive and reproduce and survive and reproduce, they can evolve into different kinds of bacteria. Early on, they would have been just eating food molecules, nutrients in their environment. We'll use this picture here to sort of show some of the different lineages of bacteria. These two down here, they make their living by eating nutrients in their environment. This one up here has evolved a primitive kind of photosynthesis. Indeed, it would go on to give rise to the more common kind of photosynthesis we see today. The green photosynthetic organisms that use carbon dioxide and water to build glucose, and oxygen is liberated into the atmosphere from that process. But we want to think about these early bacteria as pursuing successful strategies for living on Earth. They experimented with different forms of biochemistry, so they evolved into lots of different kinds of bacteria that were making a living in different ways. Then something interesting happened. If we focus on these two kinds of bacteria, scientists argue that two of them, a long time ago, some 2 billion years ago, might have merged together. Two bacteria joined together. We're going to be calling this endosymbiosis. Here you see the bacteria with the blue membrane, and then the red bacterium is inside the blue one. Now we're going to call it endosymbiosis, the symbiosis referring to two species living in close association, and the endo meaning inside or within. So this was a significant event, because after this endosymbiosis, that's when we see cells getting really large. And the idea is that endosymbiosis solved an energy problem. The little red one is today what we know as the mitochondria. It would have been really good at producing ATP molecules from the nutrients that it was getting. And the idea is, as the ancestor of the mitochondria reproduced, it gave cells a lot more energy. So cells could evolve to be bigger types of cells. So this was a really important transition in the history of life on Earth. Prior to this event, all living things were small bacteria-type cells. After this endosymbiosis event, we get the emergence of larger, more complex, single-celled organisms, which then, some of which will go on to make multi-celled plants, animals, and fungi. So let's take a closer look, though, at the relationship between these two cells that are thought to have merged in this way. Well, here we see the merger a little bit larger. We see the red bacterium is capable of dividing, but it's also really good, right, at producing ATP molecules. Well, that's a useful energy molecule that the big cell might have benefited from. So the idea is this merger between two cells was mutually beneficial. Right, so the smaller bacteria, the red one here, was producing lots of ATP for itself. But what if some of that ATP leaked out to be available to the bigger cell? Then the bigger cell would get that advantage. So the big cell got a source of ATP that was a very valuable energy molecule. Well, what about the small cell? What does it get in return? It gets protection. So the big cell is, in a sense, protecting the smaller cell, and of course providing lots of nutrients and other simple nutrient molecules that the smaller cell would have needed as well. So to say it simply, what we've got here is that the big cell benefited by getting energy, ATP energy, from the little cell, and the little cell benefited by getting protection and nutrients from the big cell. So again, let's compare sort of bacteria to complex cells. We noted that there is a size difference, and we'll see in another lesson that bacteria really face a problem. They have to remain small. The way that they produce energy, there's a certain geometry involved here. They suffer from an energy shortage as they grow bigger. So they grow bigger, but then they have to divide and remain small, and it has to do with the geometry of producing energy. Well, with endosymbiosis, this allowed larger cells to appear because inside them they could have many of these power plants, the mitochondria, powering the cell's activities. So endosymbiosis allowed cells to get big. And this too is an extremely successful strategy. So just as bacteria dominated Earth for 2 billion years, and there were lots of different kinds of bacteria, a very successful type of organism, so too these new larger cells went on to be a very successful kind of life form on Earth. So these kinds of organisms experimented with size and complexity. And of course, they went on to give rise to most of the living things that we are familiar with on planet Earth, the plants and the animals and all the macroscopic things that run around the planet. So the event that we're talking about, the first endosymbiosis, is right down here near the origin of life here. But it took 2 billion years for this event to occur. But it produced something very important: complex cells with mitochondria. So this would be the origin of complex cells, and every other thing, every other living thing on the tree of life that you see in this picture could trace its ancestry back to these first complex single-celled organisms. So if we think about the Drake equation again, we have the fraction of planets where life emerges. That's down here. But then the next term in the Drake equation is the fraction of planets that have life where the life gets intelligent. Well, it turns out then that this intelligence is really only found among organisms that are composed of complex cells. So in a sense, the origin of these complex cells was really important for the evolution of intelligent life on Earth. So we wouldn't have intelligent life on Earth were it not for the origin of these complex cells 2 billion years ago. And the shocking thing is that scientists think this was a rare event. It happened just once in the history of life on Earth. But when it did happen, it gave rise to a very promising branch on the tree of life. To say it another way, were it not for endosymbiosis, life on Earth would just be bacteria of various kinds. It was endosymbiosis that gave life on Earth the possibility of getting big, complex, and intelligent. Now let's consider another feature of some living things, that they can perform photosynthesis. And we're familiar with plants. They're multicelled, but inside the cells they have chloroplasts that capture sunlight energy to build glucose from water and carbon dioxide and release oxygen. Well, we learned that there are other organisms that are single-celled that can also do photosynthesis. Here we see euglena, and euglena are packed with chloroplasts. But we learned that photosynthesis originated in bacteria, as we saw a moment ago. So cyanobacteria are photosynthetic bacteria similar to the ones that might have lived over 3 billion years ago. But let's focus on these chloroplasts that you find in plant cells and in euglena. In fact, here's a picture of euglena on the left and then some isolated chloroplasts. One of the euglenas died and the cell ruptured, and we have a close-up view of some of the individual chloroplasts. As it turns out, then, chloroplasts also possess some characteristics that are clues to their origin. Where did chloroplasts come from? Well, consider these clues. They're the very same clues that we saw with mitochondria. Chloroplasts are roughly the same size as bacteria. They have circular DNA like bacteria, and they reproduce within the cell like bacteria. In fact, in this picture here, we see a kind of a spherical chloroplast. They're kind of disc-shaped, but this one appears to be elongated, as if it is growing, and then at some later time would divide to make two chloroplasts. In fact, in this picture, we've got two nice examples of chloroplasts in the process of dividing. So chloroplasts reproduce within the plant cell, or the euglena. Now let's remember, though, that the chloroplasts are found in these complex cells, whereas the origin of photosynthesis was cyanobacteria. So somehow we have a transition from bacteria to a complex cell with chloroplasts. Well, those three clues that we just suggested, that chloroplasts have the size, DNA, and reproduce, suggest that what happened in this transition was another endosymbiosis event. So chloroplasts were the result of a second endosymbiosis event. Somehow, a photosynthetic bacterium got inside a larger cell and stayed alive. Well, we're familiar with cells eating other cells. Bacteria, or if amoeba here is eating a paramecium, and here the paramecium is in the food vacuole. Imagine that the paramecium somehow escaped digestion and could be living in the cytoplasm of the amoeba. Here we see a stentor has eaten a euglena. Imagine that somehow that euglena remained alive inside the larger cell. Here we see a paramecium has eaten a euglena. Imagine that the euglena was not destroyed by digestion, but somehow remained alive. And so this is what scientists think happened in the origin of chloroplasts. A predatory cell ate a cyanobacteria-like organism. So in the process of feeding, it enveloped, it engulfed, and something went wrong. there was some kind of failed digestion, and that bacterium was able to survive in the cytoplasm of the cell and reproduce as it has done since its origin, right? So what we would then have is a larger cell with now a photosynthetic bacterium in the cytoplasm. This would, of course, give the big cell here a wonderful new capacity, the capacity for photosynthesis. Now, this particular kind of endosymbiosis, scientists think happened multiple times. The sort of predatory cell just eating up a bunch of stuff. Sometimes digestion failed. But the first endosymbiosis of the mitochondria is thought to have occurred only once. On our picture, then, we can represent endosymbiosis, the second one, in this manner. We have a predatory cell eating up things in its environment. It eats a photosynthetic bacterium but fails to digest. That would be the second endosymbiosis. The first endosymbiosis was the origin of mitochondria. So we have a story here then where we have large cells were only possible after the first endosymbiosis that produced mitochondria. What today we call mitochondria were once free-living bacteria. They got themselves inside a larger cell, and the combination was beneficial to both of them. So the big cell would give protection to the mitochondria, and the mitochondria would share some of the energy with the big cell. It was a mutually advantageous scenario. Some of these predatory cells are going to then go on to evolve into multi-celled animals, but some of them went through a second endosymbiosis event. They incorporated a photosynthetic bacterium, and this single-celled organism with the capacity for photosynthesis would have been the ancestors of multi-celled plants.